I was not originally supposed to be giving this talk. This talk was supposed to be given by Caitlin Gerben, who's also at the Institute, but she is off in Paris accepting her award for being the second best ultra runner in the world, which is amazing. So I am here in her stead. So as Graham talked about in his introduction this morning, one of the things that we're interested in understanding at the Allen Institute is how cells move between different states. And so what I wanted to start with is just to show you kind of what our cells look like when they are stem cells. So you can see this colony. It's kind of migratory. The cells are in a single layer. But we're really interested in understanding the process by which cells undergo this huge, crazy transformation to become cardiomyocytes. And so I hope you can immediately appreciate that the cells on the left and the cells on the right are super, super different from each other. So one of the many things that changes as cells go from being stem cells to cardiomyocytes is that they have to build this structure called the sarcomere. And so here you're looking at a diagram of some of the different components of the sarcomere. And one of the great things about working at the Allen Institute is we have access to these amazing tagged lines that are localizing to different parts of the sarcomere. So we can light those components up and actually look at how they develop and change over time. And so a lot of the work that we're going to talk about today is focused on the alpha-actinin protein specifically, which localizes to the Z disk of the sarcomere. So for reasons I'm not going to talk about in a lot of specific detail, that alpha-actinin protein is a really nice structural marker that illuminates how the organization of that organelle changes over time and kind of lets us see how that structure is being built. So there are a lot of different ways that one can approach asking these questions about how cells change and what kind of states they're in. But two ways that people in the field use are doing single-cell RNA sequencing. And so here you're looking at just a UMAP plot of some data that Rory's going to go through in a lot more detail in a few minutes. But you can see that there are many different clusters of cells that are identified by their gene expression patterns as being different from each other. And then in addition, we're an imaging institute, and so we do a lot of work looking at the actual structural changes that cells undergo as they go through these transitions. And so here you're seeing a myosin-tagged cell line that's undergoing the epithelial to mesenchymal transition. And again, you can see that there's a lot of really major structural rearrangements that are happening in those cells. And so we think that both of these kinds of changes are really important for understanding the kinds of states that cells transition through. And so we're going to talk to you today about how we're using information from both of these kinds of data to help us understand more about this specific stem cell to cardiomyocyte transition.
Great. So as Melissa said, the first thing we did to look quantitatively at the differences in these cells was running a bunch of transcriptomics on it. We assayed the cells at 4 time points, all the way from day 0, which are sort of undifferentiated pluripotent cells, all the way to day 90, which are very, very mature cardiomyocytes. And most interestingly, we have these 2 intermediate time points, which are sort of immature but committed cardiomyocytes. And those are the time points that we also have imaging data for. So we'll sort of focus in on those as we go. So when we do single-cell RNA sequencing on these cells, we get roughly 40,000 transcripts in terms of like the dimensionality of the data. We had about 20,000 cells total. This is smushing all that data down into 2 dimensions with some fancy category theory, making these UMAP projections where each dot is a cell. And the dots are placed so that similar cells in the high-dimensional space are close to each other here, while it also sort of maintains the global structure of the manifold. We can also put color on this plot. This is sort of a quick and dirty clustering in the high-dimensional space that tells us that our data is relatively sane. We see divisions roughly where we would want to see them, especially when we look at the cells colored by time point. So we see the undifferentiated cells all sort of off on their own, the very mature cardiomyocytes down at the bottom there, and the time points that we're mostly interested in up at the top, sort of less discretely distinct than the other clusters, more of a continuous blend between the 2. We can also go in and look at specific genes with some biological foresight here. So here we're lighting up the expression of SOX2, which is a pluripotency marker, and here troponin T, which is a classical cardiomyocyte marker. And you see that the day 0 cells are lit up for the SOX marker, and then there's some differential expression here of the troponin T, which is telling us that some of our cells, even within the time points, are heterogeneous, right? So some of them have differentiated what we might say correctly, and others are not, if you looked at them, going to be representative of the cardiomyocyte phenotype. We can also look at some other genes that are interesting in this intermediate zone of the day 12 and day 24 immature cardiomyocytes. And here it's really much more of a continuum. You see this MYH6 probe and MYH7, these are myosin heavy chains. And these are known biological markers of this immature cardiomyocyte state. And the cells in this process will start at the protein level to switch out from MYH6 and switch over to expressing more MYH7 as they mature. And so this lines up with what we're seeing, but it's painting a more fuzzy picture, and it's something that we're gonna want to look at more later. So remember those two genes. Okay, back to our plot here. If we zoom in on what we might say are the intermediate time points that are expressing the cardiomyocyte marker troponin T, those are sort of the cells we're interested in, finding out more about what's going on with them. We see some pretty robust transcriptomic signatures. These are some genes that are picked out as being discriminative between clusters, and it's not really super important what they are, other than that they sort of light up things that agree with what maybe a biological expert would expect. And I've highlighted here the differences between those two intermediate time points that we're interested in. We can also go in with some supervised learning. Julie has already mentioned Rob Tibshirani's lasso technique, and unfortunately we don't have the inventor of maybe the coolest statistical technique ever on our team, but we think we were okay at implementing a robust bootstrapped version of that to help us pick out genes. And honestly, it doesn't really matter the details of the method, because when you plot the genes here and look at their differential expression, on the x-axis is the mean expression, and on the y is the log fold change between the day 12 and day 24 time points. The ones that we pick out are pretty obvious ones that you might just pick out with a simple pen and paper. And specifically, two of the ones that are most prominent are these myosin heavy chains genes again. So we will come back to them later as well.
So Rory's just told you about a lot of the transcriptional differences that we see between cells that are at these day 24 and day 12 time points. But that's a period where we also see a lot of changes in the way that alpha-actinin structure is looking. And so I'm going to tell you a little bit more about how we've looked at that. So again, just to highlight here, we have this sarcomeric structure that we're looking at the maturation of in the cardiomyocytes. And one of the things that we immediately observed with this alpha-actinin is that both within a single time point as well as between time points, we actually see a lot of variation in what that alpha-actinin structure looks like and what kind of patterns we can see. So one of our kind of early approaches to looking at this was actually to go through and just manually have humans score some of the different stages of organization that we felt like we were seeing, ranging from cells that were pretty disorganized and had kind of sparse alpha-actinin and that very dot-like pattern, all the way up to cells that had these really regular structures, mostly throughout the entire cell. But one of the things that we saw there was that a lot of the cells have these kind of mixed patterns of localization. And so again, just to highlight, again, within a single time point and between time points, we can see this huge variation in the kinds of structures that alpha-actinin is forming in these cells. So given this amount of variation, we decided that we wanted to take an approach that would allow us to go through and actually classify different parts of the image as possessing different organizational motifs. And so we have these kind of 5 motifs that we felt like we were seeing really commonly in all of our data. This kind of messy pattern, these more kind of thread-like alpha-actinin patterns, random dots, and then these two more regular structures of regular dots and regular stripes. So we were lucky to have machine learning expertise in the institute, and so we were able to go through and actually train a deep learning algorithm to, on a pixel-wise basis, classify different regions of the image as possessing different organizational motifs. And so that gave us a much more detailed and finer-grained way of understanding what kind of organizational patterns we were seeing in cells. And so one of the things that, again, comes out of that analysis, being able to apply that across our whole data set, is that as cells age and are in that day 32 time point, we do end up seeing kind of more organized structures in those cells, and we see more disorganized structures at the earlier time points. And then reassuringly, all of that machine learning classification lines up nicely with how humans classified these cells. So if a person said that the cell was pretty organized, had a sort of level of 4 or 5, we see a much higher fraction of regular structures in those cells. So that gave us a lot of confidence that this measurement was picking up on something that is real in the data and something that a human observer would see in those cells as well. And so one of the great things that this kind of organizational metric allows us to do is we can actually line cells up from being less organized to more organized along this sort of pseudo-time axis. So instead of having to look at what the cell's age is in culture, we can instead just look at the organizational level of each individual cell in the data set. So we've talked about the kinds of variation now that we see in transcript expression, and then we've also talked a little bit about some of the structural variation that we've seen in our cells. And so our next step was really wanting to be able to integrate those kinds of measurements. And so to do that, we turned to RNA FISH. So just to kind of highlight what this data set is and talk a little bit more about how we looked at these kinds of variation. So we started by taking cells that were expressing the alpha-actinin 2 GFP-tagged protein, and then we performed FISH in these cells as well. And so FISH basically allows us to look at what transcripts are actually turned on in any particular cell. So once we had this data set where we had probed for 2 genes, and then in addition probed for the alpha-actinin, or looked at that alpha-actinin localization, we went through and hand-segmented individual cells. And so we had about 500 fields of view and segmented approximately 10 cells per field of view. So our data set contains just over 5,000 cells. And so what's great is that having this data set of all of these single cells allows us to go through and on a single cell level, segment probes, calculate a bunch of cell features related to the number of nuclei they have, their shell shape, as well as what that alpha-actinin 2 organization actually looks like. And so what I hope you can appreciate looking at this collection of 8 of our 5,000 cells is that, again, you can see that each of these cells looks pretty different from each other. Their shape is different. Their alpha-actinin 2 patterns are different. And then we get this really rich information because we can also look at what transcripts these cells are expressing. And again, because we get this really great spatial information, we can see that there's variation between transcripts in terms of their density, where they're localized, and their expression patterns as well. And so this is kind of showing you the panel of 8 genes that we looked at. We're going to focus a lot on this MYH6 and MYH7 pair, but we just wanted to highlight that we did look at a bunch of additional genes in this data set.
Great. So we've looked at the transcriptomics and the structural development of these cells, and then both together with the FISH probes. And so let's see what if anything, we found. Particularly, like how is the FISH probe expression, so the gene expression of this limited set of genes, related to the structural maturity of the cells? So okay, where are we coming from? We started with human expert annotations. That showed some difference, but weren't super finely resolved. We moved to an automated structural maturation scoring metric that lets us more finely discriminate these cells. And then we have gene expression measurements, and this is from the FISH data. Obviously, we have much more in the RNA-seq, but this is what we have paired with the images. And so we have these 8 probes that we selected from the strongest leads in the RNA-seq data, and then through a series of rounds of prototype FISH experiments, iterating through maybe like 30 to 50 genes, we landed on these 8 that were both differentially expressed by time point and biologically motivated, and threw in a control just for fun. Okay, and so we see some differences here by time point, nothing super stark, but some differences. But just a reminder that it's not the time point that we care about, right? It's how the structure and the gene expression correlate. The time point is just a surrogate. So let's get rid of that essentially and look at the pairwise distributions here. Okay, joint distributions. Here we have our control gene. On the x-axis, we have the fraction of the cell that's we think is a regular structure, so that's our structure metric, and on the y-axis is the gene expression, and we see essentially no correlation. That's fine, that's our control. Next, let's look at our friends, the myosin heavy chains. This is less great. There's also no correlation here, right? One would think that maybe these genes that were so statistically significant in predicting the time point, which is maybe a surrogate for maturation, would it be helpful in predicting the structural maturity of the cells? This seems to not be the case here. We can look at our other genes here, and we see the same story. The only possible exception might be this collagen gene up here. But once you control for time point, and this collagen is a marker for time point very strongly, that disappears as well. So there's essentially no correlation with structure once you control for time point across the board. Yeah, that's the one. So that structural maturity metric was just one feature. We had thousands of these features that we looked at. Here are some highlights of just the simple ones up at the top with our probe densities down at the bottom. There's some relationships here, but they mostly disappear when you control for time point again and get rid of these trivial correlations with cell area. When you do this, we don't see much, but the strongest signal is the MYH6 and 7 genes here that correlate the strongest of the genes. I'm only showing three of the gene probes here. So just to be on the safe side, we can dig deeper into this pair of genes. If you didn't catch it, when we assayed these FISH probes, we did them in pairs. We did 4 pairs of 2, and MYH7 and 6 were 1 pair. So we have these genes both in the same cell, so we can go and look a little bit deeper and construct an aggregate metric for them. So maybe individually they're not telling us something that together they might, because they're in sort of a known switching pair. So we can construct this metric where -1 is all MYH6 in a cell, +1 is all MYH7, and the transition between them. And you can see that they do segregate the cells by time point, which makes sense because we selected them because they could do that. Let's plot that against our structure metric, which again segregates. The joint distribution still is nothing there. There's higher cell density. Black was the wrong button to push. There we go. There's higher cell density here, but once you control for that, there's just no correlation. So we see that even trying our hardest with this small set of probes that were selected through a pretty rigorous process, the transcriptomics is not predictive of the structural metrics. So what are we leaving out of this? Well, we're leaving out almost entirely the spatial distribution of the probes, right? We did spatial transcriptomics. We're not looking at that. We're just aggregating the counts per cell. This is something we're excited to sort of follow up on because you do see distinct patterns depending on the genes. Something we'll work on in the future. What else are we leaving out? Well, we're leaving out essentially any sort of sophisticated analysis, sort of like maximally informative engineered features or complicated models, particularly image-to-image models. So here's an example of a very preliminary image-to-image prediction model where you just look at a non-fluorescent image of a cell and try to predict the FISH probe. You might think you should not be able to do this. It turns out that you kind of can. I'm sorry, that contrast isn't great here, but the red is the predicted FISH probe localization and the green is the actual data. And it actually lines up pretty well. This is interesting, but very, very preliminary. Okay, so what did we learn? We see really robust differences in the transcriptomics signatures of these cells and also in the structural maturity of them. And when we move to a small set of FISH genes and try our hardest to find correlation between the gene expression and the structural maturity of the cells, we just don't see it. We were not able to predict the structure from the transcriptomics or vice versa. So we need both of these modes of measurement to tell the whole story in these cells. This is just a really brief overview of this project that we've been using to look into these immature developing cardiomyocytes and the structural and transcriptomic differences that they express along their differentiation pathway. We're excited to keep going on this and get this work out to you. We spent a bunch of time developing this automated structure metric that's made the process of classifying these cells so much easier. We think that's super useful in the future and to the community. We really want to get this high quality annotated data into your hands as soon as possible. And we're also excited to broaden the scope of the FISH probe set that we're using to really get at the underlying manifold of the transcriptomics, maybe something that would really recapitulate all of the information in the single cell RNA-seq that we could have pairwise with the images. We're also looking forward to studying more interesting differentiation situations, very early differentiation in particular. And you know, in a positive light, if the data is not predictive directly of the data in the terms of transcriptomics to structure, that is kind of a call for models to bridge that time scale. And so we think there's a really interesting opportunity there. So we'd like to thank our founder, Paul Allen, for his vision, encouragement, and support. We'd like to thank the whole institute. We're mostly up at the top there. And in particular, if you have questions about the details of this work, most of these people are here and they did most of this work. So save all your hard questions for them. Thank you.
In the interest of time, we're going to take just one question, and then hopefully you can follow up with them during our coffee break.
So I guess I got lucky. The kind of obvious naive interpretation of the inability to make this correlation is that it's not, that the structure is not cell autonomous. And I got the impression from some of the early slides that you showed that in fact the structures in one cell might flank structures in another cell. So, and then you have this contraction phenomenon, right, where you'd think cells might actually work together. So have you tried sort of segmenting this, but ignoring the cell boundaries?
That's a great question. It's not something we've tried. Certainly, we have a few movies of cells and what the alpha-actinin is doing in these kind of time frames, and you can see so many dynamics between the cells and the cells pulling on each other and the beating having an effect. And so I think that's a really interesting question and something we should follow up on. I think the beauty of FISH, right, is that you do kind of get some contextual information outside of just the single cell part. So I think that's a great follow-up.
Yeah, a quick note that the very preliminary image-to-image translation is cell agnostic, and has shown some success. So it's something we're excited to follow up on in the future.
All right, thank you, Melissa and Rory.